So now we're going to go ahead and talk about the first couple steps of translation in bacteria. We're going to talk about tRNA charging, initiation, and finally elongation. So starting off with tRNA charging, we know that the goal of translation is to make this amino acid chain that'll be our protein. But our amino acids are just kind of floating around in the cytosol right now. And so we need a way for them to come together. And what we're going to use are tRNAs, because we're going to attach an amino acid to a tRNA. And that tRNA is kind of like our chaperone that's bringing all the amino acid to where it needs to be. And they are going to be very specific for each other because of an enzyme called amine acyl tRNA synthetase. There's 20 of these, and there's 20 amino acids, so there's one for each amino acid. And so it'll recognize its amino acid. So let's say glycine. It'll go to grab glycine, and then it'll look at the tRNAs and say, okay, you match up with glycine, put them together. And now that tRNA is charged or loaded, and then it can go over to the ribosome, insert it into the correct site, and start making that amino acid chain. So we're constantly having amino acids floating around, tRNAs floating around, and your amino acyl tRNA synthetase is putting them together so that we can actually make that amino acid chain. So let's look at this little image really quick, just to make sure that this all makes sense. So we're going to start by pretending that this mRNA isn't here and this amino acid isn't here. So we're just going to pretend our amino acid is completely separate from our tRNA, and they're just both floating around. So they're floating, and they're floating. Amino acyl tRNA synthetase is going to grab that amino acid, it's going to grab that tRNA, and it's going to put them together. So now we have an amino acid attached to our tRNA. Now that tRNA is going to be looking at your mRNA and trying to see where it matches up. And once it finds that spot in the ribosome, it'll be able to bond to the codon, which is what your mRNA sequence is called, because these are going to be complementary to each other. It'll be able to bind, and then that amino acid chain will start growing because of that. So that is our tRNA charging, basically taking your amino acid, taking your tRNA, putting them together so that now we can actually build that chain. So let's talk about building that chain. We're with starting at initiation. And initiation is basically just setting up our little picture to start elongation. And so we have an end goal. And so I'm going to go through this end goal, and then we're going to go through each of these, and then I'm going to draw a picture, and hopefully it makes sense. So end goal, we want 2 subunits of the ribosome with an mRNA strand between them. And we want that start codon positioned where it needs to be with the first tRNA attached to it in the P site of the large ribosome. And that first tRNA is going to be carrying an amino acid called F-methionine. So that was a lot. That's okay. We're going to break it down. First, looking at those 2 subunits of the ribosome, we have a large subunit and a small subunit, and the mRNA would be fed right through the middle. The small subunit is super special because it has a complementary sequence to a sequence on the mRNA. That sequence is in the 5' untranslated region, and it's called the Shine-Dalgarno sequence. We're going to talk a little bit more about this in a second. But big picture right here, small subunit has a complementary sequence to the mRNA, so they're able to bind together. The large subunit has 3 locations, 1, 2, 3, and they're labeled A, P, E, and these are places that your tRNA can bind. And the large subunit is also really cool in that it's actually a ribozyme, so it's a ribosome enzyme, and it can make peptide bonds between the amino acids. Now let's look at our mRNA. So mRNA, there's 3 parts: 5' untranslated region, protein coding region, and 3' untranslated region. Your 5' untranslated region has that Shine-Dalgarno sequence. This is a conserved sequence for evolution, and that's why it's able to bind to that ribosome. We want to conserve that because it allows the ribosome to recognize the mRNA for them to bind together. And in addition to just allowing that binding, it's actually also going to allow that start codon, AUG, to be put in the exact right position that we want it. So that's the 5' untranslated region. And the next one we're going to look at is protein coding region. And this is just the part that's turning into a protein. Starts with the start codon, ends with the stop codon. Everything in the middle are those amino acids. And then looking at our first tRNA, we know it's loaded with a tRNA, with an amino acid called F-methionine. And that anticodon of tRNA is able to bind to the codon AUG, which is our start codon. And when your large subunit comes in, the tRNA is going to be in the middle site, also called the P site. And finally, obviously, this is a lot of complicated things coming together, and so we need a little bit of help to do that. And those are called initiation factors. These just help bring everything together. The first two, IF1 and IF3, are going to keep the large subunit away because the large subunit is trying to bind, but we're not ready for it yet. We need to set the scene first before it comes in. That's IF1 and IF3. And then once they leave, your large subunit can come in and bind. And IF2, along with GTP, which is an energy source, is going to help bring that first tRNA over to our start codon AUG. So let's draw this really quick. So starting with our subunits, we have a small subunit, and on that small subunit, you have a sequence that's complementary to the Shine-Dalgarno sequence. And additionally, sorry, I didn't know I did that. Okay. So additionally, we have a large subunit, and it has 3 spots in it. We have the A spot. Okay, there we go. The A spot, which is all the way on the right side, the P spot in the middle, and the E spot at the end. And we're going to come to back to this large subunit in a minute, so just ignore it for now. So we have our small subunit mRNA, small subunit, and then we have our mRNA that has that Shine-Dalgarno sequence on it. So we also, on our mRNA, I'm going to go ahead and label what the protein coding region would look like. So I'm going to say it's in this section right here, and that means that AUG would be those first base pairs in that in that protein coding region. So we have those 2 green locations that are the Shine-Dalgarno sequence and mRNA, and the complementary one in the ribosome. So these are going to bind together. So we're going to bring these 2 together. And now we have that small subunit of the ribosome with the complementary sequence. And then we have our mRNA that is bound, and we have that AUG located right where we want it. So now we're going to have our tRNA, which I'm just going to draw as a T with F-methionine as the amino acid that it's charged with. And on the bottom of it, I'm going to put the anticodon, making it complementary to AUG, which would be UAC. So since they're complementary, this tRNA is going to be able to come over here and bind to AUG. So I'm going to go ahead and draw it moving over and binding there. So we have that UAC anticodon, our tRNA, and the F-methionine. All right, so now we have our first tRNA, we have our small subunit, and our mRNA. And now we need that big subunit to come over and bind to all of this. So this large subunit is going to come on over and bind to everything. And when it does that, it's going to place that tRNA that we drew in the P site, in that middle site, meaning that you have the A site and the E site still open. And that's going to be the end of initiation. So now let's go ahead and look at elongation. So elongation is all about building the amino acid sequence in order to make our protein. And our ribosome is going to be moving 5' to 3' on the mRNA. So we're reading the mRNA from 5 to 3'. And every single time the ribosome is going to move down one codon, because we know it's going to move by codons, because amino acids are based off of codons. And that's going to move the tRNA to a different site. Because the tRNA is always going to be bound to the same codon, the anticodon and codon are going to be together. Whenever the ribosome moves, the codon and anticodon stay together. And it's going to be moving to a different spot. So if it was in A, everything moves, now it's in P. But keep in mind, it's still bound to the same thing. Everything moves again, now it's in E. E is exit, where it would be kicked out. So tRNA is always moving in the direction of A to P to E, moving with wherever its codon is. So that was a lot of words that I just put up there with the example of movement. I'm going to actually draw it off to the side instead, just kind of following that format. But before I do that, just as a quick note, just like in initiation, we had initiation factors to help out. In elongation, we have elongation factors that use GTP to help the process continue. So let's go ahead and start. And I'm going to go ahead and go right back to where we were at the end of initiation, where we have our AUG, we have our tRNA, and this tRNA is bound to F-methionine. All right, and then I'm going to go ahead and add in a couple other codons. I'm going to add GGC and AAA. Okay, so now we have something in the P site, but we don't have anything in A. So we can bring in a tRNA that's loaded with an amino acid that is complementary to GGC on the anticodon. So I'm going to bring in that tRNA. It's going to come in here. It's complementary to GGC. And it is also attached to its own amino acid, which I'm just going to write AA because I'm not totally sure which one it is. So now I have something in P and something in A. And now our ribosome is going to do a super cool function and act as a ribozyme, and it's going to use its catalytic powers to make a peptide bond between what's in the P and the A for the amino acid. So now we have so many bonds in here that the P is like, I don't need to hold on to this amino acid anymore. A is taking care of it. So P is actually going to release its bond to the amino acid, meaning that now everything in the A site, or the tRNA in the A site, is holding on to the entire chain. So once this happens, now everything is going to move down one unit. And instead of actually moving my ribosome down one unit, I'm just going to pull my mRNA one codon to the side. So for example, my AUG is going to move over 1 to be in this E site. My GGC is going to move over 1 to be in the middle. And then my AAA is going to move over 1 to now be in the A site. And remember that your tRNA is going to move with your codons. So if I move my green AUG over 1, that tRNA is moving right along with it. So I'm going to move my green tRNA over to this exit site. And actually, whenever a tRNA ends up in the exit site, it's just booted out automatically. So this is actually now going to leave, and it's just going to float around, and it will end up finding its aminosyl tRNA synthetase to add another amino acid to it, and then it could come back and do the cycle again. So we just booted out of the exit site, so there's actually nothing located there right now. But we also need to move our blue tRNA over because it's complementary to GGC. So I'm going to move that over. And I'm also going to move over its little sequence that's attached to some amino acid and F-methionine. So everything has just shifted over 1. Now our A site is empty again. So we can bring in another tRNA that would be complementary to AAA. So I'm going to bring in that tRNA. And this again is going to carry its own amino acid. So now we have something in P, we have something in A. Our ribosome is going to use its super cool function and catalyze a peptide bond between the blue and the purple amino acids. And now again, the P side is like, why do I even need to hold on to this anymore? A is already taking care of it. We get rid of the bond between the tRNA and the amino acid in the P site. So now everything is hanging on to A, and then we would move everything down again. So my AUG is now out of my ribosome. My GGC is going to move over to the exit site along with its tRNA, which will hang out there for a second, but ultimately gets booted out. Draw it up there with the green one. And my AAA is also going to move over to my P site along with the entire tRNA structure. And I'm just going to extend the bond over here instead of moving all those amino acids. And then in our A site, we would have some other nucleotides, let's say CCU, and then we would have another amino acid. tRNA comes in with an amino acid that can bind to that codon of CCU, and then we just continue this process. So it's all about coming in at A, or having something in A and P at the same time. You make the peptide bond. Now A holds on to everything. We move down one. So now everything would be in P. Something comes into A, peptide bond, P lets go, A is holding on to everything. We move down one. So hopefully that process makes sense. And just as a quick mention of the last step of translation is termination. That'd be whenever you have a stop codon on the bottom, and instead of a tRNA entering, we're going to have a release factor enter, and it'll enter and just make everything fall apart and release our polypeptide chain. And that is the end of this video that goes over translation in bacteria.
I hope you found this video really helpful. The concepts and information presented in these videos will be true no matter what genetics class you are taking. However, the concepts presented in this video are referencing material currently covered in Baylor University's coursework. Remember, if you are currently enrolled as a Baylor student, we offer free tutoring services. Our tutoring center is located on the first floor of the Sid Richardson building. You will find all the details you need to know about these services on our website, www.baylor.edu/tutoring. You may schedule a free 30-minute one-on-one tutoring session online through Navigate, or just drop in during our open business hours. For more information about our current services, please visit our website. Thank you.